Today we're going to be talking about amino acids, which are the building blocks of proteins. There are 20 different amino acids, but they all have certain features in common. They all have a central carbon called the alpha carbon. They also all have a carboxyl group, a lone hydrogen, an amine group, and an R group. All of the different amino acids differ based on their R groups. Now, you will often see amino acids presented with this kind of structure, but a little closer to reality is this model here, which is showing the carbon with its double bonded oxygen and the hydroxyl group, which is part of the carboxyl group here. This is our R group, our lone hydrogen, our nitrogen, and the possibility of two hydrogens there. As we've said, all 20 amino acids have different R groups. The simplest, glycine, has a single hydrogen as its R group. The amino acids can be grouped together based on certain characteristics of their R groups. A number of them have R groups that are nonpolar, while others have polar R groups. Some are classified as acidic, while others are classified as basic. 3 amino acids, notably tryptophan, tyrosine, and phenylalanine, are classified as aromatic because of the ring structures in their R groups. The important takeaway is that every amino acid has its own distinct properties and characteristics because of its unique R group. The amino acid will have a different charge properties based on the pH of the solution that it's in. pH dictates whether the amine and carboxyl groups of the amino acid will be protonated or deprotonated. The amine group can be protonated or deprotonated as shown here. If the group has only 2 hydrogens, as we see over here, it's in its oxidized state and there is no charge. If the amine group has 3 hydrogens, as we see here, it's in its reduced state and therefore carries a charge of +1. The carboxyl group can also be protonated and deprotonated as shown here. In its reduced form, the carboxyl group has the hydrogen attached and it is neutral. In its oxidized form, the carboxyl group loses its hydrogen and it carries a negative charge. The amine and carboxyl groups of amino acids are protonated and deprotonated according to their pK values. If the pH of the solution the amino acid is in is less than the pK, the functional group is protonated. If the pH of the solution the amino acid is in is higher than the pK, then the functional group is deprotonated. Think about it this way. If the pH of the solution is low, then there are lots of hydrogens floating around, making it more likely the functional group can be protonated. The amine group has a pKa of about 9. So at a pH that's less than 9, the amine group will be protonated. And at a pH of greater than 9, the amine group will be deprotonated. The carboxyl group has a pKa of about 2. So at a pH less than 2, the carboxyl group will be protonated, and at a pH greater than 2, the carboxyl group will be deprotonated. So let's take a look at the structure of glycine, the simplest amino acid, at 3 different pHs. Keep in mind that the pKa's of the carboxyl group is 2.3, and the pKa of the amino group is 9.6. So we need to think about whether the amine and carboxyl groups will be protonated or deprotonated at a pH of 1, 6, or 13. At a pH of 1, both the carboxyl and amino groups are protonated because the pH of the solution is less than the pK for both these groups. At a pH of 6, the carboxyl group is deprotonated because its pK is 2.3. So the pH of the solution is no longer less than the pK for the carboxyl group. The amino group with a pKa of 9.6 is still protonated. At a pH of 13, both the carboxyl and amino groups are deprotonated because the pH of the solution is greater than the pKa for either of these groups. We can calculate the net charge for glycine at these different pHs. At a pH of 1, the net charge for glycine is +1 because the protonated amine group carries a charge of +1 and the protonated carboxyl group has no charge. At a pH of 6, the net charge is 0 because the amine group carries a charge of +1, but the carboxyl group has been deprotonated, so it carries a charge of -1. Thus, the net charge is 0. At a pH of 13, glycine carries a net charge of -1 because the amino group has been deprotonated. So it is now neutral, but the carboxyl group still carries a charge of -1. Let's think about what's going on at pH 6. At pH equal to 6, glycine is known as a Zwitterion, meaning it can act as an acid by donating a protein, or it can act as a base by accepting one. When the amino acid's net charge is 0, the amino acid is said to be at its isoelectric point because it would not move when it was exposed to an electric field. Every amino acid has a unique pi, or isoelectric point. The pi value for glycine is 5.97. Similar to the notion of pK values, if the pH is below the isoelectric point for a given amino acid, the amino acid will have a positive charge. And if the pH of the solution is greater than the isoelectric point for the amino acid, that amino acid will have a negative charge. Similarly, if the pH of the solution is exactly at the pi, that amino acid will be neutral. The protonation of the R group depends on the pH of the solution, just like the amine and carboxyl groups do. Aspartic acid, or aspartate, is an amino acid with an ionizable R group. The pK of its carboxyl group is 1.9, the pK of its amine group is 9.6, and the pK of its R group is 3.7. So I'd like you to test your knowledge here. What do you think the structure of aspartate would be at pH 1, 7, and 13? And while you're doing this exercise, try to figure out what the net charge would be at each pH. So pause the video, and then we'll work our way through it. All right, so here we're going to look at the structure and net charge of aspartate at 3 different pHs, 1, 7, and 13. The pK of its carboxyl group is 1.9. The pK of its amine group is 9.6. And the pK of its R group is 3.7. At a pH of 1, the amine, carboxyl, and R groups are all protonated because the pH is lower than the pKs for all of these groups. Thus, the net charge would be +1 because the amine group carries a +1 charge, but the carboxyl and R groups are neutral. At pH 7, the carboxyl and R groups would be deprotonated because the pH of the solution is higher than the pKs of both of these groups. Thus, the carboxyl group and R group each carry a -1 charge for a total of -2. The amine group is still protonated, however, because the pH is still lower than the pK for the amine group. Thus, the amine group carries a charge of +1. The net charge at pH equal to 7 is -2 + 1, or -1. At a pH of 13, all groups would be deprotonated because the pH of the solution is higher than the pKs for all the groups. The amine group is neutral, but both the carboxyl and R groups carry a charge of -1. The net charge for aspartate at pH 13, therefore, is -2. So let's try a different amino acid. Lysine has 3 ionizable functional groups with the following pK values. The amine group has a pKa of 9. The carboxyl group has a pKa of 2.2, and the R group has an amino group and therefore has a pKa of about 10.5. What I'd like you to do is to pause the video and write the equilibrium equations for the 3 ionizations of lysine. Make sure you assign the proper pKa for each ionization and show the net charge for the lysine molecule at each ionization stage. So I want you to think about the structure of the lysine molecule and its net charge, specifically at pH 9. So what is the structure and net charge of lysine at 9.0? We know the carboxyl group will be deprotonated because the pH of the solution is greater than the pK for the carboxyl group. The R group will be protonated because the pH of the solution is less than the pK. So what about the amine group? Let's take a look. Note that the pH is the same as the pK for the amino group at 9.0. So half the lysine molecules are going to have a protonated amine group and half will have a deprotonated amine group. What does that mean? When the amine group is protonated, lysine will have a +1 charge because the amine group of the R group is protonated, the carboxyl group is deprotonated, but our amine group is protonated. So +2 -1 gives us a net charge of +1. What about when the amine groups are deprotonated? In this case, the net charge on lysine is 0. Why? Our deprotonated amine group is neutral. The R group is +1 and the carboxyl group is -1. So the net charge is 0. So the answer to the question of writing out the ionization steps and the structures of lysine as we move through the different pK values and how the net charge is shown here. Since amino acids have the ability to accept donate protons, they can act as buffers. We can even draw titration curves for amino acids as you typically have done for acids and bases. Shown here is a titration curve for the amino acid glycine. At the far left of glycine's titration curve, both the amine and carboxyl groups are protonated, as we can see here. As base is added, the hydroxyl begins to deprotonate the carboxyl group. When 1/2 equivalents of OH have been added, the carboxyl groups of half of the glycine molecules will be deprotonated, while the other are protonated. At this point, the pH equals the pK value for the carboxyl group, or 2.34. After 1 equivalent of base has been added, all of the carboxyl groups are now deprotonated. At this point, the net charge on glycine is 0, and the glycine is at its isoelectric point. After 1.5 equivalents of hydroxyl have been added, half of the amine groups of glycine will be deprotonated, while the other half of the amine groups remain protonated. At this point, the pH equals the pK for the amine group, or 9.6. After 2 equivalents of hydroxyl have been added, the amine groups on all the glycine molecules will be deprotonated, and the net charge of the molecule is now -1. Notice the shape of the titration curve. The blue boxes indicate flat areas of the curve where glycine can act as a pretty good buffer within those pH ranges. Let's look at the titration curve for glutamic acid or glutamate. Glutamate has an ionizable R group. This is quite a different situation than glycine. As a result, it takes 3 equivalents of base to completely deprotonate the ionizable groups of glutamate. Notice that the 3 pK values are reached when 1/2, 1.5, and 2.5 equivalents of base are added to the solution. Notice too that the pink and blue regions of the curve are relatively flat at each of the 3 pK values. Given that each of the relatively flat regions indicates a good buffering zone, a good general rule is that an amino acid acts as a reasonably good buffer when the pH of the solution is within 1 pH unit of each of its pK values.